questions so far? Okay, so things I would like to remind you guys from last semester is I would like very much want you to participate in my lecture. Doesn't matter if it's a question or this is structured activities in class. So again, I reward that by offering 5 points. These are participation points. If you can answer quicker questions, even just one question correctly in each period of lecture, or you can answer some questions on the quiz that we're going to do, weekly quiz. Every Friday, we're going to have a quiz that's due the next Tuesday morning, 8 o'clock. And so if you can, we can get as much as 5 points for each regular exam. But I will need your participation, which means if I pose a question, I will hope you can answer me. It doesn't matter the answer right or wrong. I want to see where your thoughts are. And if you don't want to answer, then I will just wait. Okay. All right. So we go. Is this microphone sound okay? It sounds good. Okay. It sounds very echoey for me. Okay. So digestive system. This is a topic a lot of us are very familiar with, and we learned it from high school. We are very familiar with all these different portions. So a lot of things today we're going to introduce is still going to be a little bit overview and review, and so please bear with me if some of this information you haven't heard many times before. Okay, so the structure of this lecture, this session, the digestive system, is organized in 4 different proportions, and these different 4 different sections, subsections, are based on your books. So I try to follow as much as I can with regarding your book. And they are not proportional. So for example, today we hope we can finish at least 2 of these 4 sections. And so next class period after spring break, we're going to talk about starting either the regulation of the digestive system, or we're going to start with the 4 phases of integrating response to a meal. And the last one is a little bit long. We're going to talk about all the regulations of different phases after you consume a meal. Okay. So the first part, the overview of the anatomical and histological of your digestive system, and is really simple. And if you can imagine that a tube that's very, very long tube from your mouth going to the anus, and that's the whole entire length is your digestive tract. And oftentimes we call it alimentary tract, alimentary canal, and all gastrointestinal tract. Oftentimes they just call it GI. The GI is short for gastrointestinal. Okay, so the GI tract is a long tube, as we say, is connected to the external environment. And what the job is to ingest food and excrete the waste. And so this tract is separated into different distinct portions, as we described here, by those muscle rings, so we call the sphincters. Okay, so we have stomach, small intestine, large intestine, okay, forgot esophagus. So those are different portions of it. There are anatomical structures. The histological structures actually share many similar features that we're going to see in a second. There's also some accessory organs associated with our GI tract. Those are helping to releasing either enzymes or releasing bile that to aid the digestive and absorption functions. And some of those, like livers, even though liver has a huge metabolic function, we included them in the digestive function because it has a great responsibility to excrete the lipid waste. Imagine that your water-soluble waste can be excreted from urine, and your lipid-soluble waste has to be excreted from your digestive tract. So this is pretty simple. And if I ask you to locate the 6 major digestive functions to this tract, this should be pretty simple, right? Okay, so tell me very quickly, what is the main function of the stomach? It's a very, very simple. Digestion. Okay.
Mechanical processing.
What about anything else? Yes. What's that? Liquefy all the food and mechanical processing a little bit. Okay. What is the main section that is doing absorption? And the small intestine. What is the main? What is the section that is doing digestion? The main, the most of digestion are done in one portion. What do you think it is?
Colon. Colon.
Colon. Digestion. So what is digestion? Breaking down those macromolecules into smaller molecules. What do you think? What will be the portion that is most responsible for this job? Loud.
Stomach.
I heard the stomach. I also heard the small intestine. So hopefully those a little bit of split opinions will be, like united once we finish this lecture. Okay. So I'm not going to tell you right now, but you will be super clear once you've done this lecture. So imagine that if you think about the GI tract, a lot of times it's a constant moving, and it's a lot of movement, especially after you consume a meal. So if you imagine, if you don't restrain the stomach, the GI tract somehow, it's going to could potentially tangle up, could potentially cause a lot of irritations. And so there's this peritoneum is those membranes, serous membrane that helps to anchor them to the stomach wall and to the abdominal, the back of your, to your back, body wall, and to help to prevent the tangling during the movement. Okay, so I'm trying to work on the smartboard, but doesn't look to be working in this room. So I would like you to just look very carefully into this PowerPoint. And you can see some orange lining here. Okay. This orange lining that is lining against your body wall, those are the parietal peritoneum. Okay, so that's the inside the surface of the body wall. This is maybe easier to see is the blue lining here. Okay, so those blue lining here is immediately outside of your GI tract, and those are covered with this visceral peritoneum. So your peritoneum, besides those who anchors those intestines and the stomach onto the back of your wall, of your body wall, there's also these two layers here, and a parietal peritoneum and a visceral peritoneum that are constantly secreting fluid. We call it peritoneum fluid. And those fluids will help you to prevent irritations and preventing uncomfortableness when you have a very active movement of your GI tract. Okay. So in organ that is lying inside of your parietal peritoneum, that is called the intraperitoneal organs. So you can see this, all these blue lines, most of them are all in the inside of your is the intraperitoneal organs, including the stomach, the small intestine, large intestine. I will have to be careful most of the small intestine, because there is a portion of them in the first portion of your small intestine, your urine was out. So there's also retroperitoneal organs which lie in between of your parietal wall and your inner body, which is like the pancreas and also as a kidney. Those oftentimes, when they're in kind of information that give you back pain, that's the reason because of the lying right against your body wall. Okay. So here, after the broad overview of your anatomical structures, let's look at the histological structures of your GI tract. And the GI tract is very similar from portions to portions regarding these sublayers. And this is not a very totally new topic, and I know you guys talked a little bit about them during the respiratory systems. And I want to just quickly ask you, which one is the most inner layer? When I say inner layer, it's the most close to the lumen, where the food goes by.
Mucosa.
Mucosa, very good. Which one is the most outer layer? Which is close to your body, your parietal, the visceral peritoneum. Which one? Serosa. Okay. So the direction is from the most inner to the most outer layer. So let's take a look at the first inner layer, mucosa. Imagine what this mucosa really used to do. Because it's letting food go by, because our job about digestive system is mainly to reabsorb food and to reabsorb food. So the first layer has an absorption function. Okay, so those are our epithelium cells. So the epithelium cells, it could be simple columnar epithelium cells, which is most of these GI tract are covered with. Or you could be stratified if, say, you're in esophagus, which constantly has those stretch and frictions, so you need it to be stronger. So those are found in the esophagus. And then you can see that there's some type of those cells that helps to do the job. And the majority type you find in your GI tract is called enterocytes. Enterocytes, oftentimes we call it absorptive enterocytes, which is very interesting. It's actually on the cell surface membrane, which is very close to the lumen side. They express different kind of proteins. Those proteins are actually enzymes that can break down even more further the molecules into the digestible units, absorptible units that can be absorbed in. So these are the majority type. They are the ones who are responsible for moving the nutrients in. We also have the enteroendocrine cells, which are more scattered, and their job is to secrete all kinds of peptides and to modify the functions. And of course, you have those to help lubricate inside of your GI tract. You have those mucus-secreting cells, and people try to, even more confusing you guys. So they give different names for the same functions throughout the GI tract, like a goblet cell, mucus cells. So we will be. You can pretty much looking at in the same group. They are the mucus-secreting cells. Okay, once you have the food goes into your the first layer of the cell, so what they should be transported to. So you can imagine that you wanted to, most nutrient goes to the blood, so you can find the blood vessels and the connective tissues and over there. So another very important thing is a lot of those food ingested could be bacteria, could be foreign antigens that is potentially harmful for your body. So you find a lot of lymph systems there, okay, lymph nodes there. So these layers we call a lamina propria. The final layer is helping to shape your very inner mucosa. This is called vascularis mucosae. Okay, so these three layers are made of mucosa. And then you can visualize them by thinking about the function. The submucosa is a little bit away from your lumen. Okay, now these submucosa, what they need to do, where you can find them, what kind of structure you find them there? Larger, bigger vessels, and also more lymph nodes. Okay, so in fact, between the submucosa and the mucosa, you can find 50% of your lymph systems in your body. So you can find a B cell, T cell, mast cell, plasma cells, that all these, histamine, prostaglandin, all these things we have introduced in the immune system there. So it's very, very powerful, and if you think about it, they are very tolerable too. A lot of foreign food with eating, they have to be treating them like it's digestible, it's absorbable. So they're against the pathogens, against bacteria, but also will allow you to not mount an immune response to like a pork or beef that you're eating every day. Okay, so those are the submucosa. In the submucosa, we also find a nerve plexus. Here is the submucosal plexus because it's finding the submucosa. And what is the job to do is to provide the nerve supplies to the muscle layer we just talked about, the muscularis mucosa. All right, so if we're going further away from the lumen, and now we're going to the muscularis external. This is normally two-layer muscle layers. Okay, so you have a longitudinal layer outside, and you have inner is circular layers. So you can imagine in the circular layers, when it's constrict, would you see the lumen size get bigger or small?
Smaller.
Smaller, okay, not a trick question. But longitudinal, what do you think will happen? The digestive system will be shortened, right? Okay, so we're going to see why there needs to be two and why this is useful. Between these two layers, you can find another plexus, the nerve plexus, which is called a myoenteric plexus. This is the most of your nerves find in your GI tract. Very powerful myoenteric plexus, which is a part of your enteric nervous system. Go ahead.
So, what does the longitudinal layer does?
Longitudinal layers is, say this is a tubing, right? Tube. That's your GI tube. When it's contracting, it's shortening.
Okay.
Okay. So the circular layers is making it like kind of pinching on it. The longitudinal just shortening it.
Would it also widen it?
No, it won't widen it. How you're going to widen it is relaxing the circular layer. You're going to be able to widen it. So serosa. Okay. Serosa is the mosal layer, and we talk about the serous membrane, and that's part of it. So that's a connective tissue, and it has a secreting function. And the serosa becomes an adventitia when it's going to those, for example, esophagus, the pharynx, and the rectum, where you have a less of secreting job because it's less chance to get irritated and during the movement, and more of an anchoring job, and so it has a more connective tissue there. Okay, so if you take a look at this diagram, you will see that throughout the GI tract, all these different layers are preserved, but they have their kind of features, different features. For example, what is the stomach do? Stomach was basically it's a storage food, and it's secreting a lot of those enzymes and hydrochloric acid, and there's a lot of stomach fluid. So that you can see that a lot of pits, we call it pits here, it's labeled here, pits, and that has a lot of secretion function. And if you're finding the small intestine, this is not proportionally described because of the small intestine, even though it's long, the duodenum is quite short. But you can see those finger-like projections projected out into the lumen of your GI tract. Those finger-like projections are villi, and on top of them, they are microvilli. So you guys probably know those function is to greatly enhance your surface area, so you have a lot of chance for food to get reabsorbed in. Okay, so you can see that the duodenum doesn't have many villi or taller villi as the duodenum. It's because the duodenum itself during that phase is a short phase that basically to neutralize the pH and preparing the food that's coming from the stomach, so it's no longer super acid and ready for the GI, the small intestine to processing it. So you can find that they're actually quite different. It's very interesting. Okay, so this is basically the anatomical review of what we all have now from previously. It's just a quick review. And the next topic I want to not spending too much time is talking about the four major functions as your book was describing: motility, secretion, digestion, and absorption. So these four functions I'm not going to go into quite detail, because when we're going different portion of your GI tract, we're going to spend more time talking about how the motility is regulated in different portions. So at this point, we just give a very broad view of it. Okay, so first one is the motility. So motility, let's just focus on the muscularis external, which is the third layer we were talking about before. And there, in these layers, we have 2 layers in most GI tract. Some like stomach has 3 layers, but in most places we have just a circular and a longitudinal. Okay. So this might not be very obvious that those muscularis externals, different portions, they have different movement patterns. For example, they have those called tonic contractions. Tonic contractions means they contract and don't let go for quite a long time. Oftentimes, this could sustain for minutes to hours. So you imagine where you will see those type of contractions. You will see them in your sphincters, which will preventing the food to move in the wrong direction. And you will see them in the stomach, which you will want to create pressure when during the stomach emptying. Those are, we're going to see that next class will show movies to you to appreciate how tonic contraction would look like in the stomach. Okay, so most of you guys will be very familiar with the basic contractions, which is, we learned before, hopefully, peristalsis and segmentation. Are these 2 words familiar to you guys? Okay. Okay, so you can see that the peristalsis, those are lasting only a few seconds. Okay, and they have different functions. One is to moving the balls forward. That's peristalsis. And the other one is mixing the content inside, which is the segmentation. Okay. So one thing I would like to draw your attention to is if you're looking at the smooth muscle in your GI, and if you take a look at their membrane potential, they actually are oscillating very, very frequently. Okay, so they do this. They're oscillating from -80 to -40 millivolts. So very, very regular oscillation. And these patterns we can kind of see in these red trace on the top. Okay, so ignore those high peaks here. Those are the action potential. But just looking at you have those under the threshold oscillations. Those are our basic electrical rhythm. Okay, these rhythms depend on the different portion of your GI. Could range from 3 seconds every cycle to 20 seconds every cycle. So it could be very slow and it could be quite fast. So those rhythms are set by those pacemaker cells in the cell, but in the smooth muscle, and sometimes we call them ICC, the interstitial cells of Cajal, and sometimes I just call them pacesetters or pacemaker cells. And then those are found in between of these 2 layers, and those are the ICC. So, okay, so one of the things you may notice in the bottom trace here is normally those cycles does not reach threshold. If you do not reach the threshold, if you're looking at the force of the muscle contraction, you don't see contraction. Okay, so normally you don't see it. But when they were having when they're being stimulated, either by chemical, by mechanical stretch, or by vagal stimulation, and they can firing action potential, and once they pass the threshold, the action potential of firing. Depends on how strong your stimulus are, and you can having multiple action potential. So the stronger your stimuli are, and the more action potential are in one of the cycle, and you're going to generate one stronger amplitude in terms of contraction. Is it clear? Questions so far? Okay. So pretty straightforward. So I like to start with the GI tract. Okay. So you guys are familiar with the peristalsis and the segmentation. And can you told me before. So can you tell me how coordinated? Describe to me what the peristalsis does. This is a very highly coordinated movement. So what do you think peristalsis move the bolus from oral to the distal part? Yes.
It's gonna be parasympathetic followed by parasympathetic.
It is. You're saying it's a parasympathetic. Okay, so parasympathetic is
it will be followed.
Parasympathetic activation initiated can initiate the peristalsis. But what I'm trying to ask is how these muscle layers are coordinated to make this happen. What do you imagine your stomach is doing? You're just looking at the diagram is actually telling something there. Yes?
Like contracting and expanding to move the bolus down.
Okay, so contracting on the oral part of it, and at the same time relaxing on the distal part of the tube. And then also contracting your longitudinal layers that is ahead of the bolus, so make it shorter for the bolus to move for a longer distance. So with less effort, you can push the bolus much faster. Okay, so let me ask you this then. Where do you see your these peristalsis wave generally? Yes. Esophagus, okay. Esophagus is a very beautiful example because it's not the gravity that will push your food down. It's actually a very powerful peristalsis wave, actually a series of them if you got stopped, like eating an egg or something, then that's generating secondary peristalsis wave. Very good. Any other places? You guys are quiet. Any other places? What's that? Stomach. Why you need a stomach to generate peristalsis wave? Mechanical activation, that's true, but why you say mechanical breaking the food down, right? So it's okay. So you basically are generating very strong peristalsis wave to mixing the food inside of your stomach. And of course, with the sphincter closed, when you were doing this very strong peristalsis wave, you don't split anything into your small intestine, but you have a chance to mixing the food inside. Okay, so how about the small intestine? Will you see very strong peristalsis wave there? No. So normally you don't, except we call it MCC. Okay. So why you don't see very strong peristalsis wave? To me, maybe this is too obvious for you guys. To me, it's fascinating. You don't want to generating a very quick wave to push the food around, and so you don't have a time to digest and absorb the food, right? So instead, you creating it's actually, we say very weak, it's actually very minimal. That's, you see mostly is a segmentation. But the segmentation has a different frequency gradient. So because you have a different frequency, remember the contracting isolation thing, and the rhythm is different, and that will be able to push your foot down. So what you're going to see in this week's lab actually is the small intestine contracting, and very often people will confuse the saying they're looking at this peristalsis wave. It is actually segmentation. It's very, very interesting. Unstimulated intestine, and you can see that contracting very frequently. Okay, segmentation is basically contracting rings, so adjacent areas will contracting one and then the other and then the one. So it's predominantly seen in small intestine and the large intestine. Okay, I see people getting bored, so I'm going to ask you guys to do a collaborative activity and talk to your neighbor and tell me how many ways you can propose to changing a smooth muscle contractility. You can either by increase or decrease, pick one, and you can write as many as you want, and I'm going to floating around and check your answers. And 2 minutes. Okay. And this is a great time to wake up. Okay, so what you have been thinking of, not, I hope not just the, the neuromodulators outside of your cells, but also considering intracellularly those second messenger systems and the calcium, the cyclin NP, and all these involved. So hopefully we can all thinking about all these players. And in this week's lab we're going to change in different portions. So manipulating this diagram in your lab. Next week is different. Following week we're going to be manipulating those players and to see how that influences your contractility of this movement. Once you're done with it, I would like you to answer a question here. So the channel is AB in this group. And feel free to talk to your neighbor, because we do want to get at least one question right in this class period, right? All right. Okay. Going to give you 5 more seconds to click in. You haven't? All right. Okay, very good. So, can anybody defend or explain why the answer A is not correct? Yes. Yes. It's not no troponin. Where do you find the troponin? Skeletal muscle and the cardiac muscle, right? So for smooth muscle, you don't find troponin. So how does calcium work then in this case? So calcium in this case, pay attention to here. You have a calcium and a modulator. And when calcium modulating bond is actually activating those kinases, which is the calcium modulating dependent. So these myosin light-chain kinase activation will cause this phosphorus to form and activate and causing the contractility to increase. Some of you asked, answered the cyclin AMP, increased cyclin AMP. Take a look at the cyclin AMP here. It's very interesting. It's actually having an inhibition effect on myosin light-chain kinase. So that is the reason when you have acetylcholine, which is a parasympathetic neurotransmitters in the organ level, and that activating GI and causing the cyclin AMP level to drop, that will have an excitatory effect on the smooth muscle contraction. So this is a negative feedback, which means that cyclin AMP itself needs to decrease to get an excitatory effect, and acetylcholine is doing that by decreasing the cyclin AMP. And that's why parasympathetic normally has an excitatory effect compared to the sympathetic nervous system, which have a negative effect. Inhibition effect. Is there any other questions regarding this?
What about norepinephrine?
What's that?
Norepinephrine?
Norepinephrine? Yes.
What does it do?
So norepinephrine does is binding to the same receptors as we saw before, the PHGS receptors.
Okay.
So you be careful, because I didn't mention this one, is you do have different receptors to find we mentioned before have those smooth muscles, like especially vessels, glasses as we talked about. Some vessels will constrict, some vessels will relax, depends on the receptors you find. So those need to be careful of that. Some of the receptors do even though norepinephrine especially binding to it, will causing a constriction if you find that a GQ receptor is there. So that GQ receptor causes calcium to release, and that will have an excitatory effect. So that was a great question to differentiate these two. All right, so secretion. So secretion is, again, we're going to talk a few general examples, the classic examples. And the first one, starting with the oral cavity, that's the salivary glands. We have 3 salivary subtypes in there, and parotid, sublingual, and submandibular. And normally I don't ask you what those are in your exams, okay? But you know that there's 3 of them. And they are very active. So during your actively thinking about the food and your saliva is coming out, each minute your saliva is able to generate the amount of the salivary glands are able to generate the saliva at its own weight. So very, very quick. Each day you can generate 1 liter of saliva in normal, cases. So it's a very, very active gland, and then they normally generate hypoosmotic fluid, which means they have room for the solutes to come in, to be dissolved, and to be going to the GI tract. And they are heavily regulated by autonomic nervous systems. So it's almost entirely regulated just by autonomic nervous system, especially the parasympathetic nervous system. So people were talking about, if you're nervous, oftentimes you see yourself have a dry mouth. Why? Because normally your saliva is secreting constantly. When you are experiencing nervousness and your sympathetic activation go up, causing your parasympathetic to shift down, which is the seesaw we saw last semester, right? So when the parasympathetic goes down, the flow generation will disrupt it, and you can see your mouth become dry. Even though the sympathetic does have modified conversation, so it does secrete some other component, but it does not make you feel your moisture inside of the mouth. Okay, so sometimes the people may not realize that the saliva have a lot of components. They have amylase, lipase, and they have also protective mechanisms by secreting like chloride, lysozyme, and those things that to attack foreign pathogens and protecting your teeth. So if you have a problem secreting saliva, oftentimes your teeth will also be compromised as well. And the bicarb, why is secreting bicarb in your saliva? This is always interesting to me, just to see how your body can be so clever with all these little details. So imagine where the next level is going to be. It's going to be secreted, and the saliva is going to go down the esophagus and then meeting the stomach, right? So stomach has all these acid secretions. So if they got a reflux back into your esophagus does not have those protection heartburn on this problem. So bicarb is very good to neutralize those. So this is a very good, I'm always amazed by your complexity of your body, and this is one of it. And it's a very well thought for system. All right, so this is the part that oftentimes people find a little bit confusing. So I changed the slides a little bit, especially the clinical question. Hopefully it can help. So the saliva secretion, this is not on your textbook, but will be on your exam. So they secreted by those kind of the blind-ended structures, so we call it acinine. So you have an end piece here, like a balloon, and then you connect it to a duct. We call it this duct, the excretory ducts. And then you're moving into the oral cavity, okay? So the secretion of this is a very, is a two-step process. The first step, you're going to secreting, you'll call the primary secretion, and it's basically including those enzymes we talked about, and has a very similar osmolarity as your plasma. So you can find most of these ions in very similar concentration as you find in the plasma, which means the extracellular fluid. Okay, so once it goes into here, the acinar cells, we call it resembles extracellular fluid, and they go in through the duct. When they go in through the duct, ions get moved around and they shift around. So what's going to get moved into your blood is your sodium and the chloride. Okay? And when they go into what could be added back, the potassium and the bicarb, as we talked about bicarb, how important it is. And so it is going to be also a net removal of the solutes, and because it's impermeable to water, it becomes a hypoosmotic. Okay? So this is what is going to end up to have a solution is the saliva is going to, compared to the plasma, is going to be high or low in sodium. Compared to plasma. So it will be low in sodium, high in potassium, low in chloride, and a high in bicarb. Right? So we are clear here. Okay, now we're adding to the confusing part it came, I promise you guys. So now during the active chewing, what is going to happen is either active chewing or activated by seeing the food, seeing the cheesecake, and they are going to have the less ductal modification because you have a lot of food coming in from the acinar cells, and then when they're going through it, they don't have enough time to modify it, and so it ends up having less sodium to get moved back, less chloride to get moved back, less potassium to add it in. However, one thing remain high is this bicarb. Bicarb will remain high because of the actions of those secretive globules. What is this word mean? Secretive globule means something, the peptide, the amines, something that will help maintain the bicarb level high. So those are things that the modulators to, when you actively secrete saliva, those secreting also high and help to maintain bicarb high. And that's very important too, because normally during this time your gastric secretion is increased as well, and you don't want to you want to protect your esophagus. Okay, so are we clear? Did I do a good job presenting it?
Yes.
Okay, thank you guys. And now I'm going to check if I really did. All right, so answer this question, and please talk to your neighbor and to see if we can get this question clear. And one thing I want to just draw attention is now we are talking about comparing to low saliva secretion to high saliva secretion, not compared to the plasma. Okay. What is the red line represent? Which ion concentration? You can talk to your neighbor if you want. Okay, so this is the normal time is the lowest score question in the exam. So hopefully this will change this semester. And if you have a different version of the questions on your PowerPoint, that's in the past what it was showed in the exam, but it answers exactly the same question. Okay, so here is the answer. K is the one for going down, all the others still going up. Okay, so another big diagram that I want to introduce you guys is the hydrochloric acid situation. It's a classic example. And everybody who learned anatomy physiology should be quite familiar with this model. Okay, so this shows you how hydrochloric acid in your gastric glands are secreted. So how the stomach is secreting those. And first of all, I would like to just, because it is so important, I want to just join it with you guys. It's making me miss our old classroom, which we had a beautiful monitor. But what are we going to draw here is the parietal cell. So this is a parietal cell. You may be wondering why this has such a strange shape. And this is not a typical like a cell shape because of its activated state. We will see that in the future. But you can imagine that which side it will be the lumen. This will be our lumen because it has a much longer surface area. Okay. So the one I just draw here is mostly blood. So what happens is this is using your metabolic waste to generate hydrochloric acid, and how it's generated is using CO2. So CO2, water, and in those cells they express a lot of carbonic anhydrates. So Ca stands for carbonic anhydrates. So that makes your carbonic acid. And then this is what is associated into hydrogen ion and a bicarb. Okay. So the most important part of here is hydrogen ion, how it got pumped out of your cells. It is using active transporter. We call it hydrogen potassium ATPase. This is the active transporter, so it's very effectively pumping the hydrogen out. Okay, so what about the bicarb? Bicarb will move out of the cells in exchange of chloride. Because you need a chloride, right? So that's what we're going to do is to exchange bicarb and for chloride. Now chloride comes inside the cell. And how chloride leaves the cell is by channel for chloride. Okay, so there's another one that you will be I'm asking people all the time is on every single cell you have this sodium potassium ATPase. Where do you find those? In the basolateral side or in the lumen side? Basolateral side. Why? This is if you have put on the lumen, you are now going to generate the concentration gradient. When you're talking about concentration gradient, you always compare the intercellular and extracellular. You're not comparing the intercellular versus the lumen, right, which is outside of your body. So it's always on the basolateral side. And the potassium also has the channels to move out. Okay. So basically that's the most important. And when you when you quickly generate this hydrogen ion and chloride ion, you generate bicarb, and the bicarb goes into the blood, and it is actually what causes an alkaline type. changes, your plasma pH will actually elevate it after, quickly after you eat. Okay, so one of the important things that we want to remember is you can see that these hydrogen ion and the chloride did not resemble or become assembled inside of these cells. They didn't get a chance to have a high concentration intracellularly. Every time you generate this hydrogen ion, you got to be pumped out very effectively by this hydrogen potassium ATPase, sometimes we call it proton pump. So those are the reasons that we don't want these very acid compound inside of your cell, rupture the cell membrane and damage the cell. So we wanted them to be kind of assembled separate. Okay, so keep that in mind. You have hydrogen ion and a chloride separate. Okay, so another thing, if you look at your PowerPoint, the next slide is going to be bicarb secretion. So where does bicarb secrete? I want to show this one as well. So what we had before is that you see the carbonic anhydrase is very important for these type of cells. And you see the ethylene type, which is a phenomenon you see due to the generation of the hydrogen ions. Any questions? Yes.
So HCl is gonna be made in lumen?
Yes, they're made in lumen. So they are, even though they're generated, both separately generated inside of the cells, but they don't have a chance to meet and accumulate and become acid inside of the cell. Okay. So the bicarb secretion, where does bicarb secrete? We talked about the salivary gland secreted bicarb. The most bicarb actually is secreted by pancreas, and there's also secreted by small intestine. So the reason we want bicarb to be secreted is once the food enters from the stomach into the small intestine, you're going to get ready for digestion and absorption. Many of these enzymes, they do not work in a very acid environment. They have to work in a very neutralized environment. So bicarb have to be quickly secreted and be neutralized for those hydrogen ions we just generated in the stomach. Okay, so the secretion of bicarb use a very, very opposite, almost mirrored against the hydrochloric acid secretion. Yes. So we're going to draw that together. So I'm going to still using these diagrams just to show you the differences. Okay. So now I'm going to draw a cell. This cell does not find in the same area as the parietal cells. These cells is in your small intestine or in your pancreas. Okay, so this is tight junctions, you see them. Okay, so this is still the lumen side. This is still the lumen, and this is still the blood. And we say this is very mirror very well, so you still have the CO2 in the metabolic waste. These cells still express very high carbonic anhydrase, and they're still going to generate this. But now, instead of moving the hydrogen ion out, you're going to move the bicarb out, and the hydrogen ion will be moved out of the cell into the blood. Okay. So what kind of transporters are we going to see here? And we're going to see the bicarb and chloride exchange. Okay. And from the basolateral side, what you're going to see. Okay, so first we're going to see this sodium potassium ATPase, just maintaining the cell concentration gradient. For hydrogen ion, you have a sodium and the hydrogen ion exchanger. Okay, so hydrogen ion will be moved out of the cell, goes into the blood, and the sodium will come in. And you will also have another co-transporters. We call it sodium that the cells still could maintain very low sodium because of, even though you have those transporters moving sodium in, you have these sodium potassium and PPS to maintain the concentration gradient. Okay. Of course, you can see that we have chloride moves in from the axial side of the membrane to exchange the bicarb. We have a chloride moved in on the basolateral side to come in with the sodium and potassium. So there must be channels for the sodium chloride to move out. So these moving out the chloride channels are we call cystic fibrosis transmembrane conductance regulator. Okay, so in short, it's called a CFT. This is one of the channel that are mutated in the cystic fibrosis cases, and they are not functioning and are causing mucus still secreted, and the water cannot follow, and that's causing the clotting in many different areas, including GI tract, including respiration tract. Okay, so you can compare. The reason I want to draw these two together is you kind of can compare them side by side on the same orientation and to see where the channels are located. And of course, we have both our, the sodium, potassium ATPase to generating this gradient, and we have these two important channels on each side to moving for these cases, moving the bicarb out, and the other, in the earlier cases, moving hydrogen ion out into the leaf. Any questions? Yes?
When sodium chloride and potassium come in, what drives that?
What drives that? Okay, so that's the driving force is your here for your sodium. So it's actually between the concentration gradient of your sodium that generates the heat. Did I? Okay, I'm trying to. So let's go back to the projector. All right. So a couple more secretions will be finish the secretions part today. So there's many digestive enzymes, obviously, very important for digestive function to breaking those macromolecules into the absorbable unit. And so those are secreted by, as we've seen before, salivary glands. And we're going to see later is the pancreas is secreting a lot of those different enzymes. And epithelium cells in the stomach and the small intestine. The one I want to emphasize here is actually there you find in those intestinal cells that the cell membrane itself express a lot of these enzymes. And we call them brush border enzymes. And we're going to see them in the digestive function, why they are so important. They are responsible for breaking like disaccharides into monosaccharides so you can move in. And they are responsible for breaking down those oligopeptides into the tri and the dipeptides, and then so they can be transported in. So and many of those enzymes, because of they can break protein down, and they are secreted as zymogens, which is the inactive form, so precursors of these active enzymes. The biosecretion, on the other hand, is we talked about why liver is considered as a digestive system. One of the important things is the liver secreting bile. Bile is a very complex juice. And so in this bile juice you can find bile salts, which is very important for lipid digestion. And you can also find some metabolic product like bilirubin. And we also mentioned that this is very important to excrete the lipid waste as a form, for example, cholesterol. Okay. So those are very important functions for liver to secrete bile. And we're going to talk about liver many times. So here is what we're just going to limit it to, the secreting bicarb as well. And once the liver secretion of bile is a continuous event. So it's keep continuing secreting the bile in like a dripping steady speed. And once it's finished the secreting, a large amount of this bile juice into your small intestine and help to digest food. I can't talk more 5 more minutes. Okay. So digestion and absorption. Here again is an overview, a very quick overview. Digestion is basically large molecules, micromolecules into smaller units, absorbable units. And most take place in small intestine, water ions in the large intestine. So unlike what we talk about motility and secretion, motility and secretion are largely influenced by neuron input as well as the peptide hormone. And this one does not. This one really is primarily influenced by motility and the secretion of the GI. Okay, so that's quite different from what we have just seen. So if you think about the different type of the food we've ingested, they all have different fate in terms of how they get digested and absorbed. So let's take a look at the carbohydrates. So carbohydrates, the very, very important thing you need to remember is they cannot be absorbed in any of the larger forms. So they have to be absorbed in monosaccharides. Okay, so what are those monosaccharides? Glucose, fructose, lactose, 3 of them, okay? So those are very important. So how to break them down? That's 2 steps. Once you ingested a large carbohydrates, amylase will break them down into smaller portions, and you have the brush border enzymes we just talked about on the surface, expressed on the cell membrane of those enterocytes, and they're going to break it down from those disaccharides into the monosaccharides. And how could they get reabsorbed into your small intestine, into the cell? These can be viewed in 2 different ways. You can either by the transporters, you can view by different monosaccharides. We're going to see how transporters play a role here. Okay. So the way they get transported inside of the enterocytes, those are column or epithelial cells, and the way they're going to transport out of the column cells into the blood are different. And if you could look at the transporters expressed here, you have those in the first step is to going from the lumen into the cell, inside of the cell. So you find that there's a sodium glucose transporter 1. So those are the very important transporter for glucose, as well as the lactose. It's not for fructose, okay? So this is called a secondary active transporter. So why we call it a secondary active transporter? It's because we're looking at where the energy sources came from. It's actually coming from the concentration gradient of the sodium. So sodium level is high in the lumen, low inside of the cell. So the sodium comes in, brings the glucose or the lactose in. And why the sodium working concentration inside will be low? If you look at the basolateral side of the membrane, you have the sodium with the potassium ATPase, which is generating the concentration gradient. So this is our primary active transporter. That's the sodium potassium ATPase.